In analytical laboratories, reference standards are the backbone of accuracy and trust. Every assay result, every impurity value, and every potency determination ultimately traces back to a reference standard. In this presentation, we'll explore how to qualify secondary reference standards in a way that ensures both scientific rigor and regulatory compliance, in line with ICH Q2, Q6B, and Q7A guidelines. Because in the regulated world, science plus compliance equals confidence. Confidence in your data, your methods, and your decisions. Secondary reference standards quietly support almost everything we do in the lab. They ensure reliable assay results, provide consistent references for impurity testing, and help confirm identity and potency. Yet, during inspections, one question comes up again and again. How is your secondary reference standard qualified? That question isn't just about paperwork. It's about whether your analytical data can truly be trusted. When secondary standards are poorly qualified, even validated methods can produce misleading results. Let's clearly distinguish between primary and secondary reference standards. Primary standards are typically expensive, limited in quantity, and supplied by pharmacopoeias or official sources. Because of their value, they're used sparingly. Secondary reference standards, on the other hand, are qualified against the primary and used routinely for day-to-day testing. But here's the critical principle. A secondary standard is only as good as the way it's qualified. Without scientific justification, a secondary standard is simply another sample, not a reference. Regulators don't expect perfection, but they do expect scientific justification. IQ2 focuses on analytical validation, emphasizing accuracy, precision, and reliability. ICH Q6B highlights the need for well-characterized reference materials, especially critical for biotech and biological products. ICH Q7A clearly states that reference standards are GMP critical materials, requiring control and documentation. The collective message is very clear. Secondary reference standards must be qualified, controlled, and periodically re-evaluated to ensure data integrity. So what exactly is a secondary reference standard? It's a material that has been qualified against a primary reference standard and is then used routinely for analytical testing, while maintaining traceability to that primary source. Practically, secondary standards support assay testing, impurity analysis, identity verification, and potency determination. Think of the qualification process as a calibration handshake. The primary standard is essentially saying, I trust you to represent me in routine analysis. And that trust must be earned through data. Qualification is not a one-time ritual. It's a scientific life cycle approach. You must qualify a secondary standard whenever a new batch is prepared, even if previous lots performed well. Qualification is also required when there's a change in manufacturing, purification, or the primary standard lot itself. Long-term use is another critical trigger. A common lab assumption is, it worked last year, so it should work now. Unfortunately, degradation, moisture uptake, and storage conditions can quietly compromise a standard over time. Qualification starts before testing, with selection and preparation. The material must truly represent the drug substance and align with the primary standard's specifications. Purity is critical. Even small impurity differences can directly impact assay accuracy. Homogeneity is equally important. Poor mixing or small batch sizes often lead to inconsistent results. A very practical tip: prepare enough material in one well-controlled batch to avoid frequent requalification. Many reference standard failures originate from preparation, not testing. This step is the heart of qualification. The secondary standard must be directly compared to the primary standard using validated analytical methods under identical conditions. Assay, potency, identity, and impurity profiles must all be evaluated. You're not just generating numbers, you're establishing traceability. Think of this again as that calibration handshake. Yes, I trust you to represent me in routine analysis. Without this direct comparison, qualification is incomplete. Data without statistics is just noise. Qualification requires replicate testing to assess consistency and reliability. Percentage difference compares primary and secondary standards, while %RSD evaluates repeatability. Practical benchmarks, such as plus or minus 2% for assay difference and less than or equal to 2% for %RSD, are commonly used but must always be scientifically justified. The goal is not perfection. The goal is consistency with statistical confidence. This is where ICH Q7A truly comes alive. A written qualification protocol must be approved before testing begins. Upon completion, a certificate of analysis should clearly document results and traceability to the primary standard. Storage and labelling are equally critical. Temperature, humidity, light protection, assigned value, and requalification dates must be clearly defined. Remember the golden GMP rule: if it's not documented, it didn't happen. Qualification doesn't end with approval. It continues through ongoing monitoring and periodic requalification. Stability trending, result monitoring, and early identification of variability prevent out-of-trend and OOS investigations. To conclude, secondary reference standards are not shortcuts. They are controlled scientific tools. Strong qualification practices lead to confident analytics, smoother inspections, and reliable decision-making. When your reference standards are strong, your data speaks confidently during audits, inspections, and beyond. Thank you for your attention.